I turned on? Thank you very much. I'd like to thank the organizers. I'd like John Dirk and Pierre Millien and Cindy Bell for giving me this opportunity to tell you a story about some work in bioremediation that I've been engaged in for almost 20 years. So Cindy gave me this large laser pointer so I wouldn't be dyslexic in advancing slides and pointing, but I don't know if that's going to work. We'll see.
We can just do it vocally.
I can do it, right? No, back? I have to go back now. There we go. So before I go any further, I really would like to reiterate that this is a collaborative effort, and all of the work that I'm going to be describing has been in large part carried out by a fantastic team of graduate students. But as well, I've had a long-time collaboration with industry. And in fact, when I finished my PhD, I went right away and worked in consulting. So I spent 3 years of working in an industrial setting in consulting. And that's where I realized that the #1 driver at contaminated sites were these chlorinated solvents, that they are the most prevalent organic contaminants in groundwater, and I'll get into that. I'd also like to very much acknowledge collaborators from around the world, and as well funding sources, historically obviously NSERC, but I've had a lot of funding through the US, the Strategic Environmental Research Defense Program, which is operated through the Department of Defense, EPA, and DoD, that really supports a lot of work in remediation. And more lately, we've had funding through Genome Canada and the Ministry of Economic Development in Ontario, and many other organizations. Oh, geez, I knew I would do this. So these are the contaminants I'd like to talk to you about. So this compound here, perchloroethene, or tetrachloroethene, is your dry cleaning solvent. Anytime you take your clothes into the dry cleaners, unless they are using some other technique, but 98% of the dry cleaners in Toronto, we had a student survey them, still use perchloroethene. And a sister compound, trichloroethene, is the #1 industrial degreaser. And these compounds are just so wonderful for this purpose. They were developed because they don't catch fire. They are volatile, so you put the clothing into the washing machine, and all you need to do is heat the temperature up just a little bit, it turns to a gas, and your clothes are dry. And so these features of these molecules made them so widely used. And in industry, trichloroethene is the #1 degreasing agent, because anytime you have to weld metal parts together, you need to make sure they're very clean. You just dip the parts into a bath of TCE. And the parts are clean, and then you can solder them. And so this is in the semiconductor industry, in the manufacturing industry. And when these compounds were first started to use after the war, they thought they were inert. These compounds are perfectly harmless. They don't react. If you have a bottle of TCE on the shelf, you come back in a million years, it'll still be a bottle of TCE. It doesn't react with oxygen. And so these were thought to be just marvelous compounds. Unfortunately, as a result of inadvertent and sort of careless use, because nobody thought they were of any concern, these compounds have found their way into aquifers all around the world. Wherever there is an industrial operation, you'll find these compounds. And, all right. So one of the other features of these molecules is that they are more dense than water. So if you have a spill, here's a cross-section through the ground, here's the water table. If you have a spill, the contaminant sinks deeper beyond the water, it sinks down to maybe a clay lens here or somewhere that would retard the flow, and then it extends out laterally and then down further and laterally. So the extent of the contamination can be huge from one small little point source. And the contamination can be very deep. And then what happens is groundwater flows, and these contaminants are actually also hydrophobic, meaning that they're not very soluble in water. And so as the groundwater flows, you dissolve a little bit of these contaminants, but you leave behind these, what are called in the industry, dense non-aqueous phase liquids, or the undissolved pools of liquids behind. But even though they're poorly soluble in water, the solubilities are still 10,000 times higher than the drinking water limit. So the drinking water limits for these compounds have been set in the low microgram or parts per billion range. And the solubilities are in the milligram or even gram per liter range. And so you end up having a plume, a dissolved plume of contaminant that can travel very far and can impact groundwater wells downstream. And so the question is, well, how do you clean this up? And the challenge is that really because that it extends so far, because they're hydrophobic compounds and absorb onto the organic matter and onto the clay particles of the soil, that they really are hard to dislodge. It's a little bit like the oil recovery problem, getting the last drop of oil out, and it's really challenging. And I would argue that there really is not a satisfactory way or economic way of cleaning up TCE contaminated sites today. What happens generally is you install interception wells, you install wells down gradient, pump the water out, and treat the water by air stripping or activated carbon or something like that, and then re-inject the water. So there really isn't a good solution. But the ground in which all this contamination is found is not sterile. And so when I started working in this area, we started investigating the fate of these chemicals in groundwater. I mean, is there anything that happens to them? And consultants had noticed in the 70s, in the late 70s, they'd been looking at sites that were contaminated with TCE, installing monitoring wells, and realizing that there actually was some dechlorination products, that there was TCE, but some daughter dechlorination products were present. And so that began an investigation on something that I'm going to summarize now in a whole slide. So here's the parent molecule, PCE. This is your dry cleaning solvent. Here's your industrial degreaser. And what people observed was that in groundwater, particularly if the groundwater was anaerobic, if there was no oxygen in the groundwater, that you would see this reductive dechlorination process. So the green balls are chlorine, these are carbon, and it's a double bond here. And so this process was observed to occur, and it was really thought to make a bad thing worse, because vinyl chloride is even more toxic. It's a proven human carcinogen. TCE and PCE have a whole host of health effects associated with them, putative carcinogens. And so this was really thought to be a bad thing. And the word on the street was if you had a spill of TCE, it was hopefully in an aerobic aquifer, not anaerobic, because you would have this problem of formation of vinyl chloride. In 1989, researchers at Cornell University were studying the Ithaca sewage treatment plant, the anaerobic digester in Ithaca, and they found that using this digester sludge, this mixed community, they could get dechlorination of vinyl chloride to ethene. And this was a major, major landmark finding because, of course, it means you detoxify vinyl chloride to ethene. And ethene is a fruit ripening hormone. It's completely benign. And so this discovery was seminal and then unleashed a whole a suite of researchers after that trying to find out, well, how is this done? What are the microorganisms that do this? And so a whole host of research was carried out, and people were trying to understand how is this reaction happening, particularly this last one, but this dechlorination process, they knew it was mediated by microorganisms, knew it happened under anaerobic conditions, and eventually some organisms were discovered, particularly that could catalyze these first two dechlorination steps. And so the first organism that was actually isolated was this one here that I've pictured. It's called Dihilobacter restrictus. And it was found that the only things it could grow on were PCE and hydrogen. So in other words, these organisms and the whole suite of organisms subsequently are what are called organohalide respiring bacteria. So these are organisms that use the organohalide compound as we use oxygen, as their terminal electron acceptor in metabolism, and hence the term organohalide respiration. And so all of these microorganisms, Dihilobacter, Dihilospirillum, Disulfitobacterium, and so on, are strict anaerobes. They use an electron donor, often hydrogen, sometimes acetate, sometimes other things, and a chlorinated or halogenated organic molecule as their electron acceptor, on which they dump the electrons, thereby reducing these compounds. And so early research then found all kinds of organisms that could do these upper pathway, but not this lower pathway. And it wasn't until 1997, so Some 8 or 9 years later, the same group, well, Steve Zinder and others at Cornell, finally isolated the organism responsible for this transformation. They called it Dihelicocoides ethanogenes, strain 195. It's actually been renamed just this year to Dihelicocoides mccartyi in honor of an engineer, Perry McCarty, who actually brought this technology, as I will talk about later. But at any rate, Dihelicocoides seem to have the monopoly on these lower transformation processes. And so, as you can see, it took a long time to go from the process to the isolate. These organisms are very hard to grow in isolation. But fast forward now to the era of genome sequencing, and these here is a phylogenetic tree. I know you can't read it very well, but all of these genomes have been sequenced of all of these organisms, and all of them have been found to harbor a gene that is a reductive dehalogenase-like gene, or a homolog to a reductive dehalogenase. So the enzyme that catalyzes this dechlorination reaction, these dechlorination reactions are called reductive dehalogenases, and you can identify now from gene sequences organisms that harbor such an enzyme. Not sure what it might be used for, but certainly there are many, many, many in the databases. And what we can start to look at now is which organisms these are found in. So the chloroflexi are where these Dihelicocoides are found. And the chloroflexi is a deep branching, very ancient phylogeny. And so we can look at all of these organisms now and kind of think of them in two ways. That there are, there are maybe two large groups of organo halide respiring bacteria that we can get from all this genome sequencing. And this is a phylogenetic tree turned a little bit on its side so that here you have the chloroflexi, and the firmicutes are down here, and the delta proteobacteria over here. Okay, so these are different groups of bacteria. And what there seems to be is a group here, the chloroflexi, the Dihelicocoides, and Dihalogenimonas, which is another, have a very restricted metabolism. They're very difficult to grow. They need hydrogen, and they need the chlorinated or halogenated compound. They often also need vitamins and other things. And in fact, when Steve Zinder isolated Dihelicocoides strain 195, they had to add sonicated supernatant from the Ithaca sewage treatment plant to keep it going. And so, this is this group here that we're calling restricted metabolism, and their genomes also bear that out, and I'll show you in a minute. And then there's these other organisms, such as Geobacter and Anaeromyxobacterium and so on, that have a more versatile metabolism. They can use more electron donors. They can also use different electron acceptors. They're not obligate dechlorinators. They can use maybe sulfate or thiosulfate or iron or other things as electron acceptors. And so I just blew this up a little bit here to show in these restricted metabolism, these are what these organisms breathe. Every cell needs something to eat and something to breathe. So these breathe these halogenated compounds, including the chlorinated ethenes, chlorinated ethanes, chlorinated methanes like chloroform, chlorinated phenols and benzenes and benzoates, dioxins and PCBs, but they get their electrons primarily from hydrogen through fermentation in an anaerobic environment. Whilst these other organisms over here can use a wide range of halogenated and non-halogenated electron acceptors and a wider range of donors as well. So here's some pictures of Dihelicocoides. It's a very interesting organism. It's very small, 500 nanometers or less, 300 nanometers in diameter. This is the one that we study, and it's an irregular spheroid. It's actually sort of a flat, more like a blood cell type shape. And one of the things that actually is a benefit from being such a small size is that they actually travel quite well in groundwater and they can travel miles actually underground through porous medium. So I'm going to take you back now to sort of the history of my involvement in this study, in this project, or in this kind of research. As I said, after I finished my PhD, I went and worked in industry, and all of these sites so many sites were contaminated with TCE, so we just decided to get dirt and bring it back to this is in an anaerobic glove box, get dirt in a bottle, groundwater and dirt, and to see if we could enrich and isolate these organisms ourselves. And really, we went to a site where lots of ethene was detected. So when when consultants would go out and investigate a contaminated site, they'd look for PCE or TCE and all these suite of halogenated organics, but they would never really look for methane or oxygen or even ethene. These aren't contaminants, and they're not on the suite of analytical compounds that they would be targeting. So one of the first things we did was went and looked for actually these products, and lo and behold, we could see that the complete dechlorination that was observed in the Ithaca sewage treatment plant was also happening in the field. And so we just got dirt in a bottle, and we're able to enrich very quickly a consortium of microorganisms we call KB1 that rapidly takes trichloroethylene to stoichiometric amounts of ethene. And so we've had this culture in the lab for eons now, and we just feed it methanol as an electron donor. It's fermented by acetogens within the culture to hydrogen and acetate, which then hydrogen serves as the electron donor for the dechlorination. So back to the field, though, because I've been working all of these years in conjunction with my colleagues in industry, and each time they're saying, well, that's great, Elizabeth, you've got this culture that dechlorinates really well, but we go to the field, and this is what we see. We see transformation of TC to cis-DC and no further. What's the problem? And this is actually data from a bottle where we took dirt from a site where we didn't see complete transformation. And we tried all kinds of things for like close to half a year, different electron donors, adding vitamins, all kinds of things, could not stimulate dechlorination. And then finally added in the culture, and dechlorination went completely to ethene in a few days. So it really meant to us, that the limiting factor was the organism, not any kind of condition at the site. And so, of course, that made my colleagues in industry get very excited, and so we planned a very well-controlled field demonstration to prove that bioaugmentation had an effect. Because in the world of bioremediation, there's been many attempts at bioaugmenting for petroleum hydrocarbons and other things and have not been very successful. So we really needed to prove this. And so we went to an Air Force base in Texas, Kelly Air Force Base, and designed a treatment plot. This is an injection well, an extraction well, and some monitoring wells, and basically came and injected culture from an anaerobic keg, basically, with gas to inject it into the ground. And we used PCR, basically, to detect whether the organism was there and to quantify how much growth we got and also dechlorination. And so after an injection of a single 15 liters of culture, we could track the dihelicocoides in the culture at the first monitoring well after 30 days, at the next monitoring well after 74 days, and so on and so forth. And during this process, I should say that this is just 30 feet, and the groundwater flows here in a day, and it took close to 115 days for the organisms to spread. But during this whole course of 3 months, that the PCE that was at the site was completely converted to ethene. We recirculated the groundwater this way. We were able to do a mass balance and basically show that the organisms not only dechlorinated, but grew and established themselves and dechlorinated. And so my colleague, Dave Major, at Geosyntec Consultants then said, well, I think there's a business case here, and he founded Serum, that's located in Guelph, Ontario, to market this culture. And so they call it KB1 the dechlorinator, and there are all kinds of suite of molecular tools to track the organisms and the genes, the enzyme, genes for the enzymes within the culture. And they just celebrated 10 years of existence and business this year. And so this is what it looks like. The culture is injected into the ground. They're shipped in these beer kegs, as I said, that are kept anaerobically because oxygen is very toxic to the culture. The groundwater is pre-reduced by adding some cheap electron donor like molasses or lactate or food processing waste or whatever you happen to have on the site, you add to the ground. Then you inject the culture down the well. So these are some pictures. This is the production in Guelph, Ontario. They have now 100 or 1,000 liter capacity, 100 liter reactors. And they have injected probably over 500 times culture around the world now, most recently even in Europe and in Malaysia. And Canada, we're starting to use it in Canada. It was a little bit slower in Canada. So I'm going to go back to the science. So while this industrial effort was going on simultaneously and we were providing support for that effort, we were also trying to study the culture and find out more about this really intriguing genus. And so, as I said, Dihelicocoides use hydrogen, and TC is the electron acceptor, for example, dechlorinating that to ethene. And so if you're trying to grow these in pure culture, you have to provide hydrogen and TCE. It's very slow. If you grow this as a consortium, we provide actually methanol as electron donor that's fermented to hydrogen and acetate. Acetate is the carbon source, hydrogen is the energy source, and then the methanogens and the acetogens also provide vitamins that are critical to Dihelicocoides. And in the culture that we enriched KB1, it doesn't only contain Dihelicocoides, it actually contains multiple strains of Dihelicocoides that have slightly different preferences for different substrates, and contains a geobacter that's capable of dechlorinating PC to cis-DCE. And so this functional redundancy within the culture also adds to robustness. So when you're trying to use something like this in industry, the robustness is actually, or functional redundancy is really critical. Much like is in a sewage treatment plant or in our guts, redundancy is really important. And so we did clone libraries. This is the days of clone libraries, but we've also done other kinds of libraries and, as well now. But the point being is it's a very diverse community, even after 15 years of enriching on the same two substrates in batch culture. And when you look at the clone library, it's very diverse, but if you look at quantitative data, so this is using quantitative PCR, the most abundant organism is the Dihelicocoides, and you have an acetogen here, the spirochete, and a methanogen, and these are the most abundant. And the rest of all of the organisms in the culture are very low abundance. But what we observe from time to time is a flip. So spirochete, Syntrophobacter, the arrows are a little bit off, Sporomusa, Acetobacterium, and Syntrophus are all capable of fermenting methanol to hydrogen. And at times, the culture will just flip. The population will become dominated with Sporomusa, for example, instead of this spirochete. And this is, again, illustrates this diversity, functional redundancy that actually adds to the robustness and the success of the technology in the field. So finally, I'm just going to talk a little bit about the genomics and what's been learned about genomics of these strains. And so we have actually been studying not just chlorinated ethenes, but chlorinated ethanes. 1,1,1-trichloroethane was brought in to replace TCE as a solvent, thinking it would be more benign, but in fact, it is a CFC, or HCFC, so ozone-depleting substance, and moreover, it actually dechlorinates into a chlorinated ethene, so it also goes through the vinyl chloride bottleneck. And then this is another solvent that's also used industrially. And so we've completed the metagenomes of these three cultures and are still in the process and will always be in the process of analyzing these. But it has yielded a lot of information that I'll share just a little bit with you right now. 3 substrates and 3 students as well. So the hard work, especially Shriten here, has been developing algorithms for assembling metagenome data. It's very tricky to assemble metagenome data because you have clonal variations or population variations. Like I said, we have several strains of Dihelicocoides within the culture, and those multiple strains cause sequence breaks that are very difficult to resolve. So the genome sequence of Dihelicocoides ethanogenes was published in 2005, and it definitely revealed what we saw from the physiology, which is as a very small genome, 1,500 genes only, and it's highly evolved only to use halogenated organic compounds and hydrogen. So you have hydrogenases and dehalogenases, and that's it. These dehalogenases are very numerous, and many of them look to be on genomic islands or acquired by horizontal gene transfer somehow. And so if you look at a Dihelicocoides genome, as I said, that it's not a very big genome, 1,500 base pairs, and the origin of replication is up here, and these reductive dehalogenases, there's multiple different, they're all different genes, are present in large numbers relative to the size of this genome. And so a couple of things that are interesting about these reductive dehalogenases is there's a lot of work trying to understand this enzyme, because obviously it cleaves a carbon-halogen bond. One of the things that it requires is a cobalt cofactor, so a coronoid cofactor. like a vitamin B12. But the genome does not encode any of the genes for the synthesis of vitamin B12. So one of the things it absolutely relies on from its community members is vitamin B12 for the very enzyme that it needs to live, which is quite an interesting genome streamlining that's found. So if we go back to that grouping that I showed you of the versatile versus restricted organohalide respiring bacteria, here are the genome sizes. You can see that these Dihelicocoides, Dihalogenemonas, and Dihilobacter are relatively small genomes. And this is the number of reductive dehalogenase homologous genes in those genomes. And you can see that in some of these Dihelicocoides strains, there's 30 or even in 30 to 40 reductive dehalogenase homologous genes in a tiny genome. And so these organisms are niche specialists, and all they can do is dechlorinate. Whilst the more versatile organisms have fewer copies of reductive dehalogenase genes because they also have other respiratory enzymes in their genomes, and they have much larger genomes. So if we look at a maximum likelihood tree for all of the, not all, but many of the reductive dehalogenase homologous genes that we can identify in GeneBank, and they vary tremendously, and their sequence identity is, from beginning to end, is very small, but you can recognize them by, they all have a TAT export sequence at the one end, so these are exported periplasmic proteins. They have 2 iron sulfur cluster binding regions. They have a B gene that's an anchor protein that's supposed to hold the protein, and they have a putative coronoid binding domain. And so this is how we can identify them. And the interesting and the challenge here, so the color coding on this ring is the reds and the pinks are all in the chloroflexi, the blues are like Geobacter and so on, the greens are Geobacter and the blues are the firmicutes. And so you can see there is a little bit of splitting or parsing by phylogeny, although there's a group of Dihelicocoides reductive dehalogenases over here. And the stars on the outer ring are those enzymes for which we have any biochemical characterization whatsoever. So these 3 down here are the ones involved in TCE and PCE, and they're actually now biomarkers in industry. So thousands of sequences and fewer than 13 with any kind of characterization. So my take-home messages from this story are that this lucky, successful environmental biotechnology that's based on Dihelicocoides was made possible because they are obligate dechlorinators. When you inject them into the ground, all they can do is what you want them to do. They don't eat anything else. These organisms aren't created equal. They have a unique complement of dehalogenase genes, and so if they are not competent for the vinyl chloride reductase, then they won't dechlorinate vinyl chloride. And so that explains our bioaugmentation successes. They live as part of an anaerobic community, so you need the other community members are vital in providing essential nutrients that we're still trying to work out all of them. We know vitamin B12, but there are others. And so I always get this question, where do these organisms come from? Have they just grown since we've introduced these chlorinated solvents? But of course not. You can actually date the RDA's genes as to when they might have evolved, and they're much, much, much older than the chemical industry. And so these organisms surely are part of the halogen cycle on the planet. Thousands of naturally occurring halogenated compounds exist. They're formed chemically, they're formed biologically, and these organisms are the organisms that recycle these halogenated compounds. And so what does this mean as an engineer and as trying to work with contaminated sites and identify novel processes for maybe cleaning up some of these sites, or also just novel processes for synthesizing or modifying chemicals, that we need to mine the diversity. There are these organisms. Dihelicocoides was discovered a little over a decade ago. Just a few years ago, Dihalogenemonas was discovered, and I'm sure that there's a lot more dark matter, a lot more unidentified organisms in these strict anaerobic environments that we don't explore very often that can do these kinds of reactions. And then when we think about the chemical industry, we've had this experience before, but that we should make sure that chemicals look like ones that actually exist in nature. They'll be much more likely to be biodegraded than if we make something that looks completely different. So with that, I'd like to close I'd like to thank my graduate students, past and current, for all their tremendously hard work, and finally take questions maybe after the panel, but just a little plug for our center at the University of Toronto, where we are trying to integrate bioinformaticians and biologists and enzymologists into the heart of engineering. So thank you.